It sounds simple, but it's the element that makes everything possible here on Earth.
It really is the ultimate deep dive, you could say.
Mhm.
We're going from the atomic level, just carbon atoms, all the way up to the complex machines running inside your cells.
The source material for this is, well, it's pretty rigorous. It's a detailed look at the chemistry, the structure, the function of the four major types of biological macromolecules.
Exactly. And our mission really is twofold. First, we want to explore these core ideas like molecular diversity,
Mhm.
how tiny chemical changes, these things called functional groups, dictate massive biological roles.
Mhm.
And second, we're going to build up piece by piece the structure of carbohydrates,
Yeah.
lipids, proteins, and nucleic acids. We'll look at how they're made, what they do, and importantly, what happens when their structure goes wrong.
Right, because those structural failures can lead to, well, biological disasters sometimes. We're really aiming for those aha moments for you, connecting that basic chemistry right back to how our bodies actually work, or sometimes how they fail.
We absolutely have to start with why carbon? Why is it the foundation? It really boils down to two key things, versatility and stability.
Versatility and stability. Okay.
Carbon is tetravalent. That means it can form four covalent bonds at the same time.
Mm.
And these bonds are incredibly stable. That ability is what lets it be the scaffolding, the backbone for all these large biological molecules.
It's kind of unique in that way, isn't it? It can bond with itself over and over, forming these long chains, rings, branches, structures you need to build something complex.
Precisely. If carbon couldn't do that, life would probably be stuck with very small, very simple molecules. You wouldn't get the complexity we see.
So it forms the skeleton.
Exactly. But it doesn't act alone. It needs partners. The usual suspects, we sometimes use the acronym CHONPS,
CHONPS. Right. Carbon, hydrogen, oxygen.
nitrogen, phosphorus, and sulfur. Yeah. The way carbon's stable backbone interacts with these other elements with their different bonding capacities, that's what creates this almost infinite potential for different molecules, different shapes, different functions.
It's like the ultimate chemistry set, really.
Yeah.
And this ability to branch and bond in so many ways, that sets the stage for everything else, for these four big classes of molecules that make up life.
That's the foundation.
Okay, so let's unpack that first key idea, functional groups. If carbon is a skeleton, you said these groups are kind of like the personality.
Yeah, or like chemical name tags or maybe docking stations. They're small clusters of atoms attached to the carbon backbone, and they determine the molecule's chemical behavior.
So how it reacts, what it interacts with.
Exactly. And therefore, what its job is in the cell. It's amazing how these small attachments can have such huge consequences. The real power is often in these little changes.
So understanding these is crucial.
Absolutely. They define things like reactivity, solubility, whether a molecule likes water or hates it.
Hmm.
We need to go through the main ones because you'll see them pop up again and again in carbs, lipids, proteins, all of them. They tie it all together.
All right, let's do it. Where do we start?
Let's start with a really common one, the hydroxyl group. That's just AOH.
Oh, okay, like in alcohols.
Precisely. It defines alcohols. You see it everywhere in sugars, in carbohydrates. Oxygen is very electronegative, remember. So that OH bond is polar.
Meaning it likes water.
Right. It makes the molecule hydrophilic, increases its solubility in water significantly. It's a signature of water friendliness.
Got it. What's next?
The carbonyl group.
Ah.
C double bonded to O. So CO. This one is really important for sugars too.
Okay.
And here, where it sits on the carbon chain matters. If that CO is at the very end of the chain, it's called an aldehyde. But if it's within the chain, somewhere in the middle, it's called a ketone.
Aldehydes and ketones, and that matters for sugars.
Fundamentally, it's the difference between the two main types of simple sugars. Aldoses, which have an aldehyde group, and ketoses, which have a ketone group. Glucose is an aldose, fructose is a ketose. Same formula as CR, but different structure because of that carbonyl placement.
Fascinating. Okay, what else?
Next up, the carboxyl group,
Yeah.
dash COOH. Notice it's like a carbonyl and a hydroxyl stuck together on the same carbon.
COOH.
Okay, and this combination gives it its key property.
Yeah.
It's acidic. In water, it tends to donate that hydrogen ion, the H.
Ah, hence the acid part of amino acids.
Exactly. You find it in amino acids and also in fatty acids. It makes things acidic.
So if you have an acid group, you probably need a base group too.
You do, and that's the amino group, dash then HO. Nitrogen here has a lone pair of electrons, which readily accepts an cation from the solution.
So it acts as a base?
Correct. It can neutralize the carboxyl group's acidity. And yes, these two, carboxyl and amino, are the defining groups of every single amino acid. They're also important as neurotransmitters.
Right. Okay, moving on.
Let's talk about the sulfhydryl group. SHH contains sulfur. Looks simple, but it's critical for protein structure.
How so?
Two sulfhydryl groups, often on different parts of a long protein chain, maybe from cysteine amino acids, they can react together. They form a strong covalent bond called a disulfide bridge, SS.
A covalent bond. That's strong.
Very strong. It acts like a staple, locking the protein's 3D shape in place. Really important for stability.
Okay, disulfide bridges. Got it.
Then we get to a real heavy hitter in terms of energy. The phosphate group. Phosphate. Sounds familiar, like ATP.
Exactly like ATP, adenosine triphosphate.
Yeah.
This group has multiple oxygens and carries a significant negative charge, sometimes two negative charges.
That negative charge is important. Hugely. It makes it central to energy transfer. Breaking off that phosphate from ATP releases energy. Plus, adding a phosphate group to a protein called phosphorylation is like flicking a switch.
Oh, switch.
Yeah. It's probably the cell's main way of turning proteins or enzymes on or off. That big, negatively charged group changes the protein's shape and therefore its activity. It's a fast, reversible way to control almost everything.
So, thinking medically for a second, if someone has, say, a vitamin deficiency or a genetic issue that messes up phosphorylation,
Yes.
are they basically running a cell where the switches are broken? Like the proteins are there, but they can't be turned on?
That is a perfect analogy.
Mhm.
If a protein that needs phosphorylation to be active can't get phosphorylated, it just sits there, inactive. Even if the gene for it is perfect. The raw material is there, but the activation mechanism is faulty. That shows you the power of just adding or removing that one little phosphate group.
Wow. Okay. Is that all of them?
One more key one,
Right.
the methyl group. Just carbon and hydrogen.
Looks simple.
It is. And unlike the others, it's nonpolar, very hydrophobic, and generally not very reactive chemically.
So what does it do?
Its main role in biology is often regulatory, but in a different way. Adding methyl groups directly onto DNA itself, called DNA methylation, doesn't change the DNA sequence, but it can silence genes. It's a key part of epigenetics, controlling which genes are expressed without altering the code.
Okay, so methylation controls gene expression.
That's a major role. So look at what we have. With just carbon and these, 7 functional groups, we can build millions of different molecules. We've introduced polarity, acidity, basicity, ways to stabilize structure, ways to regulate activity, ways to handle energy.
All from these small chemical attachments.
Exactly. And when these different building blocks start linking together in specific ways, that's how we get our first major class of macromolecules, the carbohydrates.
All right, let's move from the attachments to the big structures. Okay, carbohydrates, sugars and starches. These are basically fuel, right? But you mentioned structure too.
They are absolutely primary fuel sources, but yes, also critical for structure. We start small with the monosaccharides, simple sugars.
Like glucose.
Glucose is the big one,
Yeah.
but also fructose and galactose. They often have the same chemical formula, C-tell-C or ORO, but the atoms are arranged differently. They're isomers. They usually form ring structures in water.
And these wink up?
They do. Through a reaction called dehydration synthesis, water comes out and a bond forms. This specific bond between sugars is called a glycosidic bond,
Glycosidic bond, okay.
and the enzymes that make these bonds are called glycosyltransferases. They're very specific. When two monosaccharides link up, you get a disaccharide.
It's like table sugar.
Exactly. Sucrose, table sugar, is glucose linked to fructose. Lactose, milk sugar, is glucose plus galactose. And maltose, malt sugar, is just two glucose units linked together.
Simple enough, but then things get bigger.
Much bigger. That's when we get to the polysaccharides. Long chains of hundreds or thousands of monosaccharides. And here's where a really crucial difference comes in.
What's that?
It's the geometry of that glycosidic bond. It depends on the form of glucose used. There's alpha glucose and beta glucose. Sounds like a tiny difference, but it dictates whether the polysaccharide is used for energy storage or for building rigid structures.
Alpha versus beta linkage determines function. How?
Let's look at storage polysaccharides first. These use alpha glucose linkages. These bonds are relatively easy for enzymes to break down, making the stored energy accessible.
Like starch in plants.
Precisely. Plants store glucose as starch. Starch comes in two forms: amylose, which is basically a long, unbranched chain of alpha glucose, and amylopectin, which is branched. It's how plants store the energy from photosynthesis.
And what about animals? We don't store starch.
We store glucose as glycogen, mostly in our liver and muscles. Glycogen is structurally similar to amylopectin, using alpha glucose, but it's much more branched.
Why more branches?
Think about needing energy quickly, like if you suddenly need to run. Those branches mean there are many ends, from which glucose units can be broken off simultaneously by the enzyme glycogen phosphorylase. It allows for rapid mobilization of glucose when blood sugar drops or energy demand spikes.
And putting it back together, when we have lots of glucose after a meal.
That's done by a different enzyme, glycogen synthase. It adds glucose units onto the growing glycogen chains.
Now, you mentioned the liver has a special role here, especially during fasting. Something about making new glucose.
Ah, yes. Gluconeogenesis. This is absolutely critical. Your brain, especially, needs a constant supply of glucose. If you're fasting or exercising for a long time, your glycogen stores run low.
So the liver steps in?
The liver kicks into high gear with gluconeogenesis, literally new glucose creation. It takes other molecules, like certain amino acids from protein breakdown, lactate from muscles, even the glycerol part of fats, and chemically converts them into brand new glucose molecules to maintain blood sugar levels, primarily for the brain.
It's a backup generator for glucose.
A vital one. It's always running to some extent, but ramps up significantly when glucose is scarce. It's a key survival pathway.
Okay, that covers storage with alpha glucose. What about the other type, the beta-glucose linkage? You said that was for structure.
Right. Structural polysaccharides use beta-glucose linkages. This geometry results in very straight, rigid chains that can pack together tightly, forming strong fibers.
Like wood.
Exactly. The major component of wood and plant cell walls is cellulose. It's just long, unbranched chains of beta-glucose. Those chains line up parallel and form hydrogen bonds between them, creating incredibly strong microfibrils.
But we eat plants. Why don't we get energy from cellulose if it's just glucose?
Here's that specificity again. Our digestive enzymes, like amylase, which breaks down starch, are shaped to recognize and break the alpha linkages. They simply cannot bind to or break the beta linkages in cellulose.
The same building block, glucose, but one tiny change in bond geometry makes one our primary food source and the other completely indigestible fiber.
Precisely. It's a perfect illustration of enzyme specificity. Cellulose passes through our digestive system as dietary fiber, which, by the way, is still important.
Right, for the gut microbiome.
Yes, our gut bacteria can often break down some fiber, and it provides bulk, but we don't get caloric energy from it directly.
Are there other structural ones?
Oh, yes. Chitin. Think exoskeletons of insects, crabs, lobsters, and also the cell walls of fungi. Chitin is basically cellulose, but with a nitrogen-containing group attached to each glucose unit. Makes it even tougher.
And bacteria.
Bacterial cell walls use peptidoglycan. It's a more complex structure, a mesh of sugars linked together with short peptides, amino acid chains. It's unique to bacteria, which is why antibiotics like penicillin target its synthesis. It weakens the wall, and the bacterium bursts.
Okay, so linking this back to health, problems with carbohydrate metabolism seem common.
Very. We have glycogen storage diseases, GSDs, where mutations in enzymes like glycogen synthase or phosphorylase or others involved in glucose processing like glucose 6-phosphatase in von Gierske's disease means glycogen can't be made or broken down correctly. This leads to issues with low blood sugar, muscle fatigue, liver problems depending on the enzyme affected.
And things like lactose intolerance.
That's a straightforward deficiency in the enzyme lactase, which breaks the glycosidic bond in lactose, milk sugar. Without lactase, the lactose reaches the large intestine undigested, bacteria fermented, and you get gas, bloating, discomfort.
And the benefit of fiber is becoming clearer, too.
Absolutely. Feeding our gut microbes is essential for overall health.
All right, let's shift gears completely. From water-loving carbs to lipids. These are the fats and oils, right? They hate water.
That's their defining feature. They are hydrophobic, water-repelling. And another crucial point, lipids are not true polymers.
Meaning they aren't long chains of repeating subunits like polysaccharides or proteins.
Correct. They're grouped together because of their shared property of being insoluble in water, largely due to being composed mainly of hydrocarbon chains or rings, but they're structurally diverse.
So what kinds are there? Let's start with fats.
Okay, fats, technically called triglycerides or triacylglycerols. These are our main long-term energy storage molecules, far more energy dense than carbohydrates.
Yeah, and their structure.
It's relatively simple. One molecule of glycerol linked to 3 fatty acid chains.
Glycerol and 3 fatty acids. And the link.
It's called an ester linkage, formed again by dehydration synthesis between the glycerol's hydroxyl groups and the fatty acid's carboxyl groups. The enzyme that breaks these bonds down, say during digestion, is called lipase.
Now, I hear about saturated and unsaturated fats all the time. What's the actual difference?
It's all in the fatty acid chains. Saturated fatty acids have no double bonds between the carbons in their hydrocarbon tails. They are fully saturated with hydrogen atoms.
Like butter, animal fat.
Exactly. Because there are no double bonds, the chains are straight and can pack together very tightly. This close packing makes them solid at room temperature.
And unsaturated.
Unsaturated fatty acids have one or more double bonds in their tails. Where you have a double bond, you have fewer hydrogen atoms.
Plant oils, right. They're usually liquid.
Yes, and crucially, most naturally occurring double bonds are in a cis configuration. This creates a distinct kink or bend in the fatty acid chain.
A kink.
Yeah, a bend. These kinks prevent the chains from packing closely together. They push each other apart, which is why unsaturated fats, like olive oil, are liquid at room temperature.
Okay, so cis kinks mean liquid. What about trans fats? They sound bad.
They generally are. Trans fats are typically created artificially through a process called hydrogenation, where unsaturated oils are treated to make them more solid, like for margarine. This process can convert the natural cis double bonds into trans double bonds.
And trans bonds don't kink the chain.
Correct. Trans fatty acid chains are relatively straight, more like saturated fats. They can pack together, but biologically they behave differently and are strongly linked to increased risk of cardiovascular disease, raising LDL, bad cholesterol.
So fats are for energy storage. What other lipids are vital?
The absolute superstars of structure are the phospholipids. They form the basis of all cell membranes.
How are they different from triglycerides?
They start with a glycerol backbone, like fats, but they only have two fatty acid tails attached. The third position of the glycerol is attached to a phosphate group, which is negatively charged and often has another small polar group attached to it as well.
So two fatty acid tails and a phosphate head.
Exactly, and this structure gives phospholipids their unique, almost magical property. They are amphipathic.
Amphipathic.
Means they have two personalities towards water. The fatty acid tails are strongly hydrophobic, water-fearing, but the phosphate group head is highly polar and charged, making it strongly hydrophilic, water-loving.
A water-loving head and water-hating tails. What happens when you put them in water?
Spontaneous organization. To escape the water, the hydrophobic tails cluster together, pointing inwards. The hydrophilic heads orient outwards, facing the water. The most stable arrangement they form is the lipid bilayer.
Two layers. Tails in, heads out.
Precisely. And this lipid bilayer is the fundamental structure of every cell membrane and the membranes around organelles inside eukaryotic cells. It creates a barrier, separating the inside from the outside, controlling what gets in and out. It's the boundary that makes a cell possible.
That's incredible. It just happens automatically because of the chemistry.
It's a beautiful example of self-assembly driven by basic chemical properties, mainly the hydrophobic effect. And these membranes aren't rigid walls. They're dynamic.
The fluid mosaic model.
That's the one. The lipids and embedded proteins are constantly moving, shifting. It's fluid. And that fluidity is actually regulated by another type of lipid.
Which is?
The steroids.
Steroids, like cholesterol. Or hormones.
Both. Chemically, steroids are quite different from fats or phospholipids. Their structure is characterized by 4 fused carbon rings. They're still lipids because they are largely nonpolar and hydrophobic.
And cholesterol is the main one. It gets a bad rap sometimes.
It does, but cholesterol is absolutely essential. It wedges itself into the phospholipid bilayer of animal cell membranes.
Why?
It acts like a fluidity buffer. At warm temperatures, like body temp, it actually restricts the movement of phospholipids, making the membrane less fluid and less permeable. But at cooler temperatures, it disrupts the tight packing of phospholipids, preventing the membrane from solidifying. It keeps the fluidity just right across a range of temperatures.
So it maintains membrane integrity.
Yes, and perhaps even more importantly, cholesterol is the starting molecule, the precursor, from which your body synthesizes all the vital steroid hormones.
Like testosterone and estrogen.
Testosterone, estrogen, progesterone, cortisol, the stress hormone, aldosterone, regulates salt balance. All of these crucial signaling molecules are derived from cholesterol.
Wow. Okay, so medical connections for lipids seem obvious too. Heart disease.
Definitely. Dyslipidemia's abnormal levels of cholesterol or triglycerides in the blood are major risk factors for atherosclerosis, the buildup of fatty plaques in arteries, leading to heart attacks and strokes. Our understanding of steroid hormones is fundamental to endocrinology, reproductive medicine, anti-inflammatory treatments.
And problems with phospholipids.
Yes, defects in the metabolism or breakdown of lipids, especially complex ones found in nerve cell membranes, can lead to severe inherited conditions called lipid storage diseases. Tay-Sachs disease is a tragic example, where a specific lipid accumulates in brain cells due to a faulty lysosomal enzyme causing progressive neurodegeneration.
Okay, we've done fuel, carbs, and boundary storage, lipids.
Yeah.
Now the real workhorses of the cell, proteins.
Absolutely. If you want something done in a cell, catalyze a reaction, transport a molecule, build a structure, fight an infection, replicate DNA, chances are a protein is doing it. They're incredibly diverse and perform almost every task.
And their building blocks are the amino acids. How many are there?
There are 20 standard amino acids used to build proteins in virtually all life forms.
And they all have a similar basic structure.
Yes, every amino acid has a central carbon atom, called the alpha carbon. Bonded to it are four things: an amino group, NH-rose, a carboxyl group, -rho, COOH, a hydrogen atom, beta H, and then the variable part, the side chain, designated as the R group.
Ah, the R group. That's what makes each of the 20 amino acids different.
Precisely. The R group determines the amino acid's unique chemical properties. Is it nonpolar and hydrophobic, polar and hydrophilic? Is it acidic with another carboxyl group, or basic with another amino group? This variety in R groups is key to protein diversity.
And they link together to form chains.
Right. Amino acids are joined by peptide bonds formed between the carboxyl group of one and the amino group of the next, again through dehydration synthesis. A chain of amino acids linked this way is called a polypeptide.
Okay, here's where you said it gets really interesting. The function of the protein depends entirely on its 3D shape, and that shape comes from different levels of structure.
Exactly. This is absolutely fundamental. Structure dictates function. A polypeptide chain is just a string until it folds into a specific, stable, three-dimensional conformation. If that folding goes wrong, the protein usually can't do its job, and sometimes it becomes actively harmful. There are four levels we talk about.
Level 1.
Primary structure. This is simply the linear sequence of amino acids in the polypeptide chain. A, B, C, D. Just the order. It's determined directly by the genetic code in the DNA. Changing even one amino acid in this sequence can sometimes have drastic effects down the line.
Okay, the sequence. Level 2.
Secondary structure. This refers to regular repeating patterns of folding in localized regions of the polypeptide backbone. It's not about the R groups yet, just the backbone atoms.
What kind of patterns?
The two most common are the alpha helix, which is a coiled, spring-like shape, and the beta sheet, where segments of the chain lie parallel or anti-parallel to each other, forming a pleated sheet structure.
And what holds these shapes together?
Hydrogen bonds. Specifically, hydrogen bonds forming between the CO group of one peptide bond and the NH group of another peptide bond further down the chain for alpha helices, or on an adjacent strand for beta sheets. It's backbone-to-backbone interaction.
Got it. Alpha helix and beta sheet, held by H bonds in the backbone. Now, level 3.
Tertiary structure. This is the overall, complex, three-dimensional shape of a single polypeptide chain. This is where the interactions between the variable R groups come into play, causing the chain with its alpha helices and beta sheets to fold up into a specific, compact, often globular shape.
So the R groups are driving this level of folding. What kinds of interactions?
All sorts, reflecting the diversity of the R groups. You have hydrogen bonds between polar R groups. You have ionic bonds between positively charged basic and negatively charged acidic R groups. You have the crucial hydrophobic interactions.
Hydrophobic interactions.
Yes. The nonpolar hydrophobic R groups tend to cluster together in the core of the protein, trying to get away from the surrounding water. This hydrophobic effect is a major driving force in protein folding. Think of oil drops coalescing in water.
Okay, and didn't we mention those disulfide bridges earlier?
Absolutely. The disulfide bridges, SS, those strong covalent bonds formed between the sulfhydryl groups of two cysteine amino acids, are also part of tertiary structure. They act like strong staples, permanently locking parts of the folded chain together.
So tertiary structure is the final 3D shape of one chain, stabilized by R group interactions and disulfide bonds. This folding process sounds incredibly complex. Does it ever go wrong?
It does, and it's a huge problem. That's why cells have helper proteins called chaperone proteins or chaperonins.
Chaperones, like at a dance.
Sort of. As a polypeptide chain is being made on the ribosome, or if a protein gets stressed by heat, these chaperones bind to it temporarily. They shield the sticky hydrophobic regions and help guide the protein along the correct folding pathway, preventing it from clumping together improperly or misfolding.
They prevent aggregation. Okay, is there a fourth level?
Yes, quaternary structure. This level only applies to proteins that are composed of more than one polypeptide chain, subunit. It describes how these multiple subunits associate and arrange themselves to form the final functional protein complex.
Can you give an example?
Hemoglobin, the protein that carries oxygen in your blood, is a classic example. It's made of 4 individual polypeptide subunits, 2 alpha chains, and 2 beta chains that have to fit together perfectly to function correctly. Collagen, the structural protein, is another example, formed from 3 intertwined helical chains.
So primary is sequence, secondary is local coil sheets, tertiary is the 3D fold of one chain, and quaternary is the assembly of multiple chains.
Got it. Now, a huge number of proteins are enzymes, right?
A massive number. Enzymes are biological catalysts. They speed up chemical reactions, often by millions or billions of times, without being consumed in the reaction themselves. They do this by lowering the activation energy needed to get the reaction started.
And they're specific, highly specific. Each enzyme typically catalyzes only one or a very small number of related reactions. This specificity comes from the unique 3D shape of its active site.
The active site.
Yes, a specific pocket or groove on the enzyme surface where the reactant molecule, called the substrate, binds. The shape and chemical environment of the active site are precisely complementary to the substrate, like a lock and key, or perhaps more accurately, like a glove fitting a hand, the induced fit model.
Do enzymes need anything else besides the protein chain?
Many do. They often require non-protein helpers called cofactors or coenzymes.
What's the difference?
Cofactors are typically inorganic substances like metal ions, think magnesium, MbGO, zinc, D and GRU, iron, POR. Coenzymes are organic molecules, often derived from vitamins. Things like NADO and FAD, which are crucial for energy metabolism, are coenzymes derived from niacin and riboflavin B vitamins.
So the protein part alone might not be enough.
Correct. The protein part of such an enzyme is called the epoenzyme. It's inactive. When the epoenzyme binds its necessary cofactor or coenzyme, it becomes the complete active enzyme, called the holoenzyme.
Okay, so this connects back to vitamins again. If you lack a certain B vitamin,
Yes.
you might not be able to make the active holoenzyme for critical metabolic pathways. Your apoenzymes are sitting there, but they can't function because they're missing their coenzyme partner.
That is exactly right. Many vitamin deficiency symptoms arise precisely because key enzymes cannot function without their vitamin-derived coenzymes. The apoenzyme protein is synthesized just fine based on the gene, but the catalytic machinery is incomplete without the coenzyme. It highlights the interdependence of these different molecular components for overall system function.
Which makes the whole process seem fragile. What happens when folding goes badly wrong? You mentioned it can be toxic.
Terribly toxic. Misfolded proteins don't just fail to do their job, they often tend to stick together, forming aggregates or clumps that can disrupt cell function and lead to disease, particularly neurodegenerative diseases.
Like Alzheimer's.
Alzheimer's is a key example, characterized by the buildup of beta-amyloid plaques, aggregates of a misfolded protein fragment outside neurons, and tau tangles, aggregates of a misfolded cytoskeletal protein inside neurons. Parkinson's disease involves aggregates of a protein called alpha-synuclein. But perhaps the most bizarre and frightening example is prion diseases.
Prions. Those aren't viruses or bacteria, right? Just proteins.
Just proteins. It's astonishing. A prion is essentially an infectious, misfolded version of a normal cellular protein called PRPC, prion protein cellular. The misfolded infectious form is called PRPSC, prion protein scrapie, after the sheep disease.
How can a protein be infectious?
This is the terrifying part. When the misfolded PRPSC encounters a normal PRPC molecule, it somehow induces the normal protein to refold into the abnormal PRPSC conformation.
It converts it, like a zombie protein.
That's a common analogy. This conversion sets off a chain reaction. One PRPSC makes another, those two make two more, and so on exponentially. These misfolded proteins then aggregate into clumps that kill nerve cells, leading to devastating brain damage.
Creating holes in the brain.
Yes, that's why they're called transmissible spongiform. Encephalopathies, they make the brain look spongy. Examples include Creutzfeldt-Jakob disease, CJD in humans, mad cow disease, BSE in cattle, and scrapie in sheep, all caused by a misfolded protein propagating itself.
It really underscores how critical that precise 3D folding is. Okay, proteins do the work, but they need instructions. That brings us to the fourth and final class, nucleic acids, DNA and RNA. The information molecules?
Exactly. They store, transmit, and help express hereditary information. They are the blueprints and the messengers.
And their monomers are called nucleotides.
Right. The monomer unit is the nucleotide, and each nucleotide itself is made of 3 components joined together.
3 parts to a nucleotide. What are they?
A 5-carbon sugar, a phosphate group, the same one we discussed earlier, and a nitrogenous base.
Okay, sugar, phosphate, base. Let's start with the bases. That's where the information is stored, right? The ACGT.
That's the code, yes. The nitrogenous bases come in two chemical families. The purines, which have a double ring structure, adenine A and guanine G,
A and G are purines.
and the pyrimidines, which have a single ring structure, cytosine C, chymine T, and uracil U.
CTU are pyrimidines. Wait, T and U.
Right. Thymine T is found almost exclusively in DNA. Uracil U is found almost exclusively in RNA. C is in both.
Okay, so bases are A, G, C, T in DNA and A, G, C, U in RNA. What about the sugar part of the nucleotide?
That's the other key difference between DNA and RNA. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose.
Deoxyribose versus ribose. What's the difference?
It's tiny but significant. Deoxyribose is missing one oxygen atom on its second carbon atom compared to ribose, deoxy. This small change makes DNA chemically more stable than RNA.
Which makes sense if DNA is the long-term storage molecule.
Precisely. RNA is generally more involved in temporary roles like carrying messages, so slightly lower stability isn't as big a disadvantage and might even be beneficial for turnover.
Okay, so nucleotides link up to form DNA and RNA. Let's talk about DNA structure, the famous double helix.
Yes, the structure solved by Watson and Crick in 1953, building on work by Rosalind Franklin and others. DNA typically exists as two long polynucleotide strands wound around each other to form a double helix.
Like a twisted ladder.
Exactly. The sugar-phosphate components form the backbones, the sides of the ladder. The nitrogenous bases pair up in the middle, forming the rungs.
And the strands run in opposite directions.
They do. They are anti-parallel. One strand runs in the 5' 3' 3' direction, and the other runs 3 to 5 rho. This directionality is based on the numbering of carbons in the sugar ring and is critical for how DNA is read and copied.
And the base pairing, the rungs of the ladder, is very specific, isn't it?
Extremely specific, and this is the key to DNA's ability to store and replicate information. Adenine, A, on one strand always pairs with thymine, T, on the opposite strand.
A with T.
And guanine, G, always pairs with cytosine, C.
T with C.
This is called complementary base pairing. A purine always pairs with a pyrimidine, keeping the width of the helix uniform.
What holds these pairs together?
Hydrogen bonds. 2 hydrogen bonds form between A and T. 3 hydrogen bonds form between G and C.
3 bonds between G and C, only 2 between A and T. Does that matter?
It does. DNA regions rich in GC pairs are held together more tightly than regions rich in AT pairs because of that extra hydrogen bond per pair. They require more energy, more heat to separate.
Which scientists actually use, right? Like in PCR.
Absolutely. Techniques like PCR, polymerase chain reaction, used constantly in labs to amplify DNA, rely on carefully controlled heating cycles to separate the DNA strands, denaturation. The temperature needed depends directly on the GC versus AT content of the specific DNA sequence being targeted. The basic chemistry dictates the lab protocol.
Amazing. Okay, that's DNA. What about RNA? You said it's usually single-stranded.
Generally, yes. RNA is typically a single polynucleotide strand, though it can fold back on itself to form complex 3D structures by base pairing within the same strand, like A with U, G with C, and its roles are much more diverse than just storing information.
Like carrying the message from DNA.
That's a major one. Messenger RNA, mRNA, is transcribed from a DNA gene sequence and carries that code out to the ribosomes, where proteins are synthesized.
In ribosomes themselves.
Ribosomes are made of ribosomal RNA, RNA, complex with proteins. RNA forms the structural core of the ribosome and even acts as the catalyst, a ribozyme, for forming peptide bonds during protein synthesis.
What else? There's tRNA too, isn't there?
Yes, transfer RNA, tRNA, is the crucial adapter molecule. It reads the 3-letter code words, codons, on the mRNA and brings the corresponding specific amino acid to the ribosome to be added to the growing polypeptide chain.
So mRNA is the message, RNA is the factory, tRNA is the delivery truck bringing the parts.
That's a good analogy. And we're discovering more RNA roles all the time, especially in regulation.
Regulatory RNAs.
Yes. Small RNA molecules like microRNAs, miRNAs, and small interfering RNAs, siRNAs, can bind to specific mRNA molecules and either block their translation into protein or mark them for destruction. It's another layer of controlling gene expression after the gene has been transcribed.
So nucleic acids aren't just static blueprints, they're incredibly dynamic information processing systems.
Absolutely. Which brings us to what happens when the information itself gets corrupted. Mutations.
Errors in the DNA sequence. Given how precise protein folding needs to be, this must be a big deal.
It can be catastrophic, although many mutations have no effect, and evolution actually relies on mutation as a source of variation. Cells have sophisticated DNA repair mechanisms, but they aren't perfect.
What kinds of mutations happen?
The simplest are point mutations, affecting just a single base pair. This could be a substitution where one base is swapped for another.
Does that always cause a problem?
Not necessarily. Because the genetic code is redundant, multiple codons can specify the same amino acid. A substitution might result in the same amino acid being incorporated that's a silent mutation.
Okay.
But if the substitution changes the codon to specify a different amino acid, that's a missense mutation.
Like sickle cell disease. I remember reading about that.
That's the classic textbook example. A single point mutation in the gene for the beta-globin subunit of hemoglobin changes one glutamic acid, which is hydrophilic, to a valine, which is hydrophobic. This one change causes the hemoglobin molecules to clump together under low oxygen conditions, distorting the red blood cell into that characteristic sickle shape, leading to all the symptoms of the disease. It's a dramatic consequence of a tiny change in primary structure.
Wow. One amino acid.
And the third type of substitution is a nonsense mutation, where the change creates a premature stop codon, telling the ribosome to stop translation early. This usually results in a truncated, non-functional protein.
Okay. Substitutions. What else?
You can also have insertions or deletions of one or more nucleotide pairs. If the number of bases inserted or deleted is not a multiple of 3, it causes a frame shift mutation.
Frame shift.
Remember, the genetic code is read in 3-letter codons. Inserting or deleting 1 or 2 bases shifts that reading frame for the entire rest of the gene. All the codons downstream from the mutation will be wrong, leading to a completely different and usually non-functional amino acid sequence. Frame shifts are often devastating.
And there are bigger mutations, too.
Yes, large-scale chromosomal mutations can happen, where large segments of chromosomes are duplicated, deleted, inverted, or moved to different chromosomes, translocations. These often have severe consequences for development or can contribute to cancer.
Cancer seems tightly linked to mutations.
Fundamentally, cancer arises from an accumulation of mutations in genes that control cell growth, division, and DNA repair. Mutations in tumor suppressor genes or oncogenes disrupt normal cell cycle control, leading to uncontrolled proliferation.
And viruses. They mutate rapidly.
Especially RNA viruses, like HIV and influenza. RNA replication machinery is often less accurate than DNA replication, leading to higher mutation rates. This allows these viruses to quickly evolve and evade our immune system or antiviral drugs, which is why we need new flu vaccines every year.
But our understanding of DNA is also leading to new therapies, isn't it? Like CRISPR.
Absolutely. CRISPR-Cas9 gene editing technology is a direct result of understanding how bacteria use RNA guides and DNA-cutting enzymes, like Cas9, to defend against viruses. We've harnessed that system to allow us to make precise, targeted changes to DNA sequences in living cells with relative ease. It holds incredible promise for correcting genetic diseases, though there are significant ethical considerations too. So if we step back and look at the whole picture, what we've really seen today is how complex life emerges from relatively simple chemistry, starting with carbon.
Right. Those functional groups provide chemical identity.
Which leads to the diversity of the 4 macromolecule classes. And notice how crucial those weak, non-covalent interactions are hydrogen bonds, hydrophobic interactions. Individually, they're weak, but collectively they stabilize huge, complex, yet dynamic structures, like the DNA double helix or a perfectly folded protein.
And it's the interplay of all these polymers, carbs, lipids, proteins, nucleic acids that enables everything. Metabolism, heredity, cell structure, communication.
Exactly. And studying this complexity often involves 2 different perspectives: reductionism versus a systems view.
Reductionism being breaking it down.
Yes, the traditional scientific approach. Isolate one component, one molecule, one reaction, and study it in detail. For instance, figuring out exactly how the enzyme DNA polymerase adds one nucleotide to a growing DNA strand in a test tube. It's essential for understanding the fundamentals.
But that's not the whole story in a living cell.
No. The systems view tries to understand how all the components interact within the network. DNA replication isn't just polymerase. It involves helicases to unwind the DNA, primase to start synthesis, ligase S to join fragments, proofreading enzymes or pair enzymes, proteins that regulate when replication happens. It's a whole coordinated machine.
And understanding the system is crucial for diseases like cancer.
Cancer is the perfect example of a systems failure. It's rarely just one broken part. It typically involves mutations in multiple pathways, pathways controlling growth, pathways controlling cell death, apoptosis, pathways involved in DNA repair, pathways that allow cells to invade tissues. The failure of the network leads to the disease.
It really highlights the complexity, and we wouldn't know any of this without some key historical breakthroughs, right? People who figured this stuff out.
Absolutely. We stand on the shoulders of giants. You have to go back to Friedrich Wöhler in 1828.
Willer, what did he do?
Before him, there was this idea of vitalism, that organic compounds found in living things possessed some special life force and could only be made by living organisms. Wöhler synthesized urea, a known organic compound from urine, using only inorganic starting materials in his lab.
He made an organic molecule from non-living stuff.
Yes, it shattered the vitalism theory and essentially founded organic chemistry, showing the same chemical laws applied to living and non-living matter.
Huge step. Who else?
Emil Fischer, around the turn of the 20th century. He did incredible work on the structure and stereochemistry of sugars, figuring out their 3D shapes. And he also proposed the famous lock and key model for enzyme substrate specificity, a concept still fundamental today.
Lock and key. Still hear that.
Then in the mid-20th century, you have the giants of molecular biology. Frederick Sanger was incredible. He developed methods to determine the amino acid sequence of proteins, first insulin, and later, methods for sequencing DNA itself. He won 2 Nobel Prizes for this work. His sequencing techniques opened the floodgates.
And Watson and Crick, of course.
Watson and Crick in 1953, figuring out the double helix structure of DNA, based heavily on the X-ray diffraction images produced by Rosalind Franklin. That structure immediately suggested how DNA could store information and replicate.
And more recently, those prions.
Yes, Stanley Prusiner. He doggedly pursued the idea, against much skepticism, that the infectious agent in diseases like Scrapie and CJD was solely a misfolded protein with no genetic material involved. He coined the term prion and eventually won the Nobel Prize in 1997 for proving it.
It's amazing how these discoveries build on each other, and the medical relevance just touches everything we've discussed.
It really does. Carbohydrates linked to diabetes, metabolic syndromes, GSDs. Lipids are central to cardiovascular disease, hormone therapies, membrane disorders. Proteins explain countless enzyme deficiencies, inherited diseases like sickle cell, and the neurodegenerative prion in Alzheimer's diseases. And nucleic acids are the basis for understanding genetic disorders, viral infections, cancer genetics, and now revolutionary tools like CRISPR.
This has been a massive journey, starting from just carbon's ability to form 4 bonds, all the way up to intricate protein machines and the elegance of the DNA double helix. It's clear that the entire structure and function of life hinges on the chemistry, the specific shapes, and the dynamic interactions of these 4 classes of molecules. We kept seeing that specificity, A pairing with T, G with C, alpha versus beta glucose linkages determining digestibility. One wrong amino acid causing sickle cell chaos.
That absolute reliance on specificity and precise structure is really the core message. Which leads me to a final thought for you to ponder. We've talked about the incredible complexity needed for a protein to fold correctly, sometimes needing chaperone helpers. We talked about the high fidelity needed for DNA replication with proofreading and repair systems. Considering all the intricate machinery required just to maintain these complex polymers once life exists, think about the monumental challenge, the sheer chemical improbability and ingenuity required for the very first self-replicating polymer, the precursor to life itself, to arise spontaneously from the non-living chemistry earlier. Given the fragility and complexity we discussed today, what strikes you most about that first leap into existence? It really started as carbon business partners, somehow finding